Hello students, in this video we are going to study about fat metabolism which occurs by beta oxidation pathway. So beta oxidation pathway. Right. So fats are broken down into glycerol and fatty acid by the activity of the enzyme lipase. Now this glycerol, by undergoing through a series of reactions, it will be converted into DHAP, dihydroxyacetone phosphate. This dihydroxyacetone phosphate undergoes isomerization into 3-PGAL, 3-phosphoglyceraldehyde. This 3-phosphoglyceraldehyde enters into glycolytic pathway through which it undergoes complete breakdown through the aerobic respiration. Now fatty acid. This fatty acid undergoes conversion into acetyl coenzyme A through this beta oxidation pathway. Now this acetyl coenzyme A enters into Krebs cycle where it undergoes complete breakdown. So now let's study the beta oxidation pathway in which this fatty acid undergoes conversion. Its conversion into acetyl coenzyme A. Okay. So first of all, before the entry of beta oxidation pathway, this fatty acid undergoes activation into acyl coenzyme A. So this is activation. This activation occurs inside the cytoplasm of the cell. So this activation occurs inside the cytoplasm of the cell. So inside the cytoplasm of the cell, fatty acid undergoes activation into acyl coenzyme A. During this activation, one molecule of coenzyme A is added to this fatty acid and it consumes ATP, resulting in the release of AMP. AMP is released from ATP, means here two molecules of ATP is consumed during the activation of fatty acid into acyl coenzyme A. So let's say the fatty acid is palmitic acid, which is 16 carbon fatty acid. Now it is converted into acyl coenzyme A called palmitoyl coenzyme A by undergoing activation inside the cytoplasm. Now from cytoplasm, this acyl coenzyme A, that is palmitoyl coenzyme A, enters into mitochondrion where it undergoes beta oxidation into acetyl coenzyme A, after which this acetyl coenzyme A enters into Krebs cycle for its complete breakdown. Right. Now this acetyl coenzyme A, after entry into mitochondrion, it enters into beta oxidation. So at first it undergoes, after entering into mitochondrion, it undergoes dehydrogenation or oxidation. So into a compound called X. I am giving these compounds as X, Y, Z. I am not giving the much details of this beta oxidation pathway. I am giving main steps of this beta oxidation pathway. Right. So acyl coenzyme A undergoes oxidation into a compound called X. During this oxidation or dehydrogenation, what happens? Protons and electrons are released. These protons and electrons are picked up by FAD, so that FAD is converted into FADH2. It is reduced into FADH2. Now, this compound called X undergoes oxidation, undergoes hydration into a compound called Y. Hydration is nothing but addition of water. Now this compound called Y undergoes again oxidation or dehydrogenation into Z compound. So when it undergoes oxidation or dehydrogenation, the protons and electrons which are released from this compound Y are picked up by NAD+, so that NAD+ is reduced into NADH plus H+. Now this Z compound undergoes breakdown, during which before breakdown, one molecule of coenzyme A is also added to this Z compound and it undergoes breakdown, during which one molecule of acetyl coenzyme A is released from this Z compound. So this acetyl coenzyme A is with how many carbons? 2 carbons. And what will be left now? Acyl coenzyme A, which is with 14 carbons. Actually we started this pathway with a fatty acid containing 16 carbons. So when 2 carbon compound is released from the 16 carbon fatty acid, what happens? 4 carbon fatty acid, that is acyl coenzyme A, palmitoyl coenzyme A actually, but now acyl coenzyme A is remaining. So this is about the complete one complete cycle of beta oxidation. So let's see what is formed by the end of one complete cycle of beta oxidation. How many acetyl coenzymes A's are formed? So by the end of one cycle of beta oxidation, what is formed? One acetyl coenzyme A. And how many NADH2s are formed? One NADH plus H plus is formed, and how many FADH2s are formed? One FADH2 is formed by the end of one cycle of beta oxidation. One cycle of beta oxidation. Right. So now acyl-CoA. This acyl-CoA having 14 carbons again undergoes one more cycle of beta oxidation. Again and again it undergoes beta oxidation cycle until it gets completely broken down into acetyl coenzyme A molecules. So it undergoes how many cycles again? Actually 7 cycles. 7 cycles. It is 16 carbon compound. For its complete breakdown, how many cycles of beta oxidation occurs? Means 7 cycles. Because in the 7th cycle, in the last cycle, 2 molecules of acetyl coenzyme A will be released. By the end of first cycle, 1 acetyl coenzyme A is released and 14 carbon fatty acid is left. This 14 carbon fatty acid enters into second cycle of beta oxidation, by the end of which 1 acetyl coenzyme A is released and 12 carbon fatty acid will be left, and it again enters into third cycle of beta oxidation by the end of which 1 molecule of acetyl coenzyme A will be released, and what will be left? 10 carbon acyl CoA will be left. Like that 10 carbon acyl CoA again enters into beta oxidation pathway inside the mitochondrion itself. Like this acyl coenzyme A undergoes beta oxidation pathway again and again until it gets completely broken down into acetyl coenzyme A molecules, which enter into Krebs cycle, where they undergo complete breakdown with the formation of energy, that is ATP. So this is 16 carbon fatty acid. It undergoes 7 cycles of beta oxidation, because in the 7th cycle, that is in the last cycle, it forms 2 acetyl coenzyme A molecules. So by the end of 7 cycles, how many acetyl coenzyme A molecules will be formed? 8. Because per one cycle, 1 acetyl coenzyme A molecule. But in the last cycle, that is in the 7th cycle, 2 acetyl coenzyme A molecules will be formed. So 7 plus 1. Next, how many molecules of NADH plus H plus? 7 molecules of NADH plus H plus, because per one cycle 1 NADH plus H plus. And how many molecules of FADH2? 7 molecules of FADH2, because per one cycle 1 FADH2. Now when one molecule of acetyl CoA undergoes Krebs cycle, breakdown through Krebs cycle, how many ATPs will be formed? 12 ATPs will be formed. We already studied in Krebs cycle. Per one turn of Krebs cycle, 12 molecules of ATP. Per 2 turns of Krebs cycle, 24 ATP. So here 12 ATP. 12 ATP per 1 acetyl CoA. So per 8 acetyl coenzyme A, 12 ATP. So when 1 NADH undergoes oxidation through ETS, how many ATPs? 7 into 3 ATPs. Next, FADH2, 7 into 2 ATPs. So here 104 ATPs, here 21 ATPs, here 14 ATPs. So totally 131 ATPs. It comes out to be 131 ATPs. But among these 131 ATP, 2 ATPs are utilized during the activation of this fatty acid. So 131 minus 2. So what is the net gain of ATP? By the oxidation of one molecule of palmitic acid, that is fatty acid, is 129 ATP is the net gain. 129 ATP is the net gain. So like this, we have to calculate the number of ATP formed from a fatty acid. So to calculate the number of ATP formed from a fatty acid, we can use formula. So to find the number of acetyl coenzyme A, we can use the formula N by 2. N by 2. Because where N equals to number of carbon atoms present in that fatty acid. So 16 carbons, 8 acetyl coenzyme A's are formed. So to find the number of NADH, we can use the formula N by 2 minus 1, where N is representing the number of carbon atoms present in that particular fatty acid. Again, in case of FADH2 also, N by 2 minus 1. So By using these formulas, let's find the number of ATP formed by the breakdown of a fatty acid. So there is a fatty acid called arachidinoic acid. Arachidinoic acid. So number of carbons present in this fatty acid, arachidinoic acid, is 20 carbons. So what we have to find? We have to find the number of acetyl coenzymes formed from this fatty acid by beta oxidation cycles, and then number of NADH2 and then number of FADH2 formed. To find the number of acetyl coenzyme molecules, what formula we have to use? N by 2. N by 2, where N is representing the number of carbon atoms present in that particular fatty acid. So 10 acetyl coenzyme A molecules. 10 acetyl coenzyme A. Next, coming to find the number of NADH, N by 2 minus 1. So N by 2, 10. 10 minus 1, 9. 9 NADH. 9 NADH. Next, to find the number of FADH2 also, N by 2 minus 1. So again 9 FADH2. So 10 acetyl coenzyme. For one acetyl coenzyme, how many ATPs? When it undergoes complete breakdown through TCA cycle, that is Krebs cycle, 12. So 10 into 12, 120 ATP. Here 9 into 3, 27 ATP. And here 9 into 2, so 18 ATP. So we have to add all these ATPs, and then we have to subtract 2 ATPs which are utilized during the activation of this arachidinoic acid. That comes out to be the net gain of ATP, which are formed due to the oxidation, due to the complete breakdown of this fatty acid called arachidinoic acid. So this is all about fatty acid metabolism, especially beta oxidation pathway. So you understood this concept. I hope you understood this concept. By using these formulas, try to solve the questions based on this fatty acid metabolism. Thank you, students.